So what we're going to do in this problem is find the charge of the peptide species at a certain pH. Now, before we do that, we need to understand how this is different from finding the charge on an amino acid. In an amino acid, you have an alpha carbon, right? A carboxyl group, an amino group, right? And then our R group. And then the hydrogen doesn't really contribute anything. Now, the amino group can contribute a positive or neutral charge. The carboxyl group can contribute a negative or neutral charge. And then the R group can differ, right? If you have an acidic hydrogen on the R group, it can probably provide a negative charge, a positive charge, or a neutral charge. It just depends on the R group. Now, some groups, R groups don't even have an acidic hydrogen, so they can't even contribute a charge at all. Now, if this amino acid is in the middle of a sequence for a peptide species, right? Like, for example, over here we have histidine, alanine, tryptophan, aspartic acid. Like, they're in the middle, right? They have their amino group and their carboxyl group occupied being in a peptide bond, right? So now they can no longer work on being acidic or basic, right? They can't contribute a charge anymore. The only thing that's contributing a charge to an amino acid in the middle of a sequence is the R group. Now, if this amino acid is at the beginning of the sequence, like cysteine over here, the amino acid is free, but the carboxyl group is not. And so they call that the N-terminus. Now, on the other hand, if they're at the end of a sequence, right? If at the end of the sequence, like for example in lysine, the carboxyl group is what's free. And so now you have a carboxyl group and an R group, and they call that carboxyl group the C-terminus, right? So overall, the things that are going to contribute a charge to a peptide species are the R groups in every single one of these amino acids, and the C-terminus that's going to end up over here, and the N-terminus that's going to be over there, right? So this is N-terminus, and the C-terminus is going to be at the end. Now, we also have to find out the pKa's of each of these R groups, right? So for cysteine, for example, we have a thiol group, and the pK of that is 8.3. For histidine, we have a carbon followed by an imidazole group, right? And over here in this imidazole group, when it's protonated, has a positive charge on it. But meanwhile, in cysteine, when it's protonated, it does not. And the pKa for this is 6.0. Now, alanine does not have acidic hydrogen, neither does tryptophan, but aspartic acid does. Aspartic acid does have a carboxyl group on it, alright? And the pKa for this is 3.9. For valine, there is no acidic hydrogen, and for lysine, we do have a basic group, right? We have our amine at the end, when protonated, has a positive charge, and the pKa for this is 10.5, right? And so now we have all the pKa's that we need to find out what the charge on this species is, right? So now we're going to list them. We're going to put them in a list from least to greatest just for sake of order, right? So we're going to have the C-terminus, right, which has a pKa of 2.2. We're going to have our aspartic acid carboxyl R group, right? So that's going to be 3.9. And then we have our next highest, which is histidine, at 6.0. We have our cysteine at 8.3. We have our lysine group at 10.5. And then we have our N-terminus at 10.8, right? And we're going to compare that to the pH that we have. So the pH that we have is of 7.8, right? So pH of 7.8 is a lot higher than the pKa of the C-terminus, which means comparatively this is a basic pH for the C-terminus. And so the C-terminus is going to be deprotonated and provide a -1 charge. And it's going to be the same thing for aspartic acid. Now, not the aspartic acid amino acid, but the aspartic acid R group. Remember, the aspartic acid carboxyl and amino groups in the sequence is being occupied for the peptide bonds. Now, for histidine, it's a little different. When histidine is deprotonated, it has a neutral charge, not a -1. So you need to keep track of that. Make sure you don't give it a -1 just because it's less than the pH. Now with cysteine, it's a little different. Cysteine has a difference of 0.5 from the pKa and the pH, right? 8.3 minus 7.8 is 0.5. That's less than 1. And so we're going to have to use the Henderson-Hasselbach equation for that to figure out the proportions, right? Because it's not completely one species or the other in terms of the charge. Now with lysine, it's a greater difference than 1, and lysine, when it's protonated, is going to have a +1 charge, and same thing with the N-terminus. And so if we cancel out these charges, all that's left that's going to determine the charge is the cysteine group, right? And so if we just use the Henderson-Hasselbach equation, where we put the pH equal to the pKa of that group, plus the logarithm of the concentrations of now cysteine with a negative charge, when it's deprotonated, it has a negative charge, versus the concentration of cysteine when it has a zero charge when it's protonated, right? And what we find that when we solve for this, we find that we have a 74% majority of the cysteine with a zero charge compared to the 26%, right, of cysteine with a negative charge. And this kind of makes sense because the pKa of cysteine is higher than that of the pH, right? So it's going to remain mainly protonated, which is this group right here. However, since it's close, it's a really small difference between the pK and the pH, so that of 0.5, which is less than 1, you are going to find groups of it, like some partial group of it, that is going to be deprotonated and have a negative charge. And so, since all the other charges canceled out and histidine is 0, the only thing that's determining the charge of the entire species is that of cysteine. And so we say that 74% of the peptide species has a 0 charge. However, we also say that 26% has a negative 1 charge on it, right? And so that is how you find the charge of the peptide species.